Well, good morning, everyone. It's always a little intimidating to be the last lecture before lunch, so hopefully I can keep your attention and as you're thinking about what you're going to be eating. And I want to just thank the organizers of the meeting and Dr. Barr for selecting this abstract, and also my wife, Erin, who's in the audience, who listened to a few earlier tortured versions of this talk. I think hopefully it's been polished since then. So in the spirit of the theme of the conference, what does it mean to be human? What I'd like to talk about is ancestry mapping and some of the work I do and others, and try to weave it into this theme. So I want to start off with, and just as a disclaimer, I'm a genetic epidemiologist. I'm not a philosopher. I'm not an archaeologist or linguistic. So I'm an applied scientist. I just want to put that disclaimer out there. But I'm going to be brave here and talk a little bit about our Aristotelian philosophy and the four essential elements, which you can read up here. But as human individuals, as people made in the image of God, we have a fifth essential question. Who are you? And this is a question we all ask ourselves at some point in our lives and continue to ask ourselves. And it's an essential question of who our being as human beings. And this question can be in part addressed by ancestry. And we actually find in the Bible, two of the four gospel writers attempt to address the question of who Jesus is by tracing his ancestry. Luke traces it all the way back to Adam, and Matthew traces it back to Abraham. Both of them trace it through David, and signifying the kingship of Jesus. But if you look more closely at this ancestry and how the two gospel writers trace it, you'll find that the line from Jesus to David varies significantly between the two gospel writers. There's really nothing in common between the two. So how is that? Well, this just goes to show you that ancestry mapping is a very inexact science. And so here you have two very gospel writers getting to the same conclusion in terms of David being an ancestor, but in a totally different way. So no talk about ancestry and genetics would be complete without talking about the father of modern genetics, Gregor Mendel. And you all know through your high school genetics that Mendel had his P experiments, which gave us the essential laws of genetic segregation. And these are still used today in terms of thinking about ancestry mapping. I found this paper recently, which I thought was kind of interesting. This was a commentary on a distant relative of Mendel, Father Clemens Richter, who also happened to be an Augustinian monk. Dr. Richter was related to Mendel through his sister's line, about 4 generations removed. And I want to just read briefly from this commentary because I think it gives us a sense of Mendel, the person, and a little bit of pride that he might have had in his work, but the importance of the science in his work. And so I quote from Father Richter, Mendel considered the results and impact gained from his experiments in plant hybrids as fundamental. A few months before his death, he told a novice in the monastery, Franz Barina, who later became his successor as abbot, even though I have experienced some dark hours during my lifetime, I am grateful that the beautiful hours have outweighed the dark ones by far. My scientific work has brought me great joy and satisfaction, and I am convinced that it won't take long that the entire world will appreciate the results and meaning of my work. Well, as most of you probably know, Mendel's work was not realized in his lifetime in terms of the importance, but it was later. But this goes to show you a little bit about the man and the pride he took in his work. And so modern-day ancestry mapping from the lay individual is probably most known by all these different companies now that are taking a little bit of your spit in a cup and they're sequencing your genome. And it's just really striking how popular this has become. I found this article recently. 26 million people have submitted their DNA for ancestry typing. And you can see just the explosion of this in the last couple of years in terms of its popularity. So there's something about us as human beings where our ancestry is important. We want to know more about where we came from in terms of our ancestors. And our genetics is one way to get at that question. If you submit your DNA to one of these companies, and this is an example for 23andMe, this is what you get back. Depending on where your ancestry is from, they'll show you the different country of origins where your DNA maps to. And along your chromosomes, they'll actually do what's called a chromosome painting. Well, they'll able, and I'll talk about this a bit more, how this is done later, might talk about how they can actually tell the different segments of DNA and their ancestral origin. And then they'll also give you some inference about when these different ancestral origins came into your ancestry, tracing back hundreds of years. But buyer beware, this is not always, the results you get back are not always correct. And these two young ladies, two identical twins, did a little experiment recently where they sent their individual DNA into 5 different ancestry companies. And when they got back the results, lo and behold, even though they have identical DNA, they did not get identical results back. Why is this? Well, part of it's just because of the probabilistic models that are used to do this ancestry mapping. And you can see, particularly for the smaller ancestral groups here, like the broadly European, there's wide differences in terms of what these companies said that was part of their ancestry. So it's just making the point here that this is an inexact science. It's based on probability. It's based on incomplete data. And so even when you have the same raw material you're starting off with, you can end up with different results. Well, you saw this graph from our last speaker, and I'm not going to belabor it, but just the idea that human beings, the thought right now, Homo sapiens have come out of Africa and migrated through the world. But one way we know that, and we can show that, is through modern genetics. And so in the last 10 to 20 years, there's been an explosion of genotyping of individuals from different parts of the world. Whole genotyping where we have a complete genetic picture of all different populations across the world. And what this shows us is that genetic diversity is greatest in the African continent. And as you move outside of the African continent, that genetic diversity tends to diminish to the point where you get to the Americas and the native people of the North and South America, that genetic diversity is least. And so this is a way we infer this actual migration out of Africa. And so based on these genotyping and genetics, you can infer different ancestral groups, and you can break these groups up into many smaller pieces as possible, but for the purposes of a lot of the work we do, roughly about 6 major ancestral categories or groups seem to exist. Those individuals from Africa, those individuals from Europe, the Middle East, Southeast Asia, East Asia, Oceania, and Americas, and then there's a small group here in Central Asia called the Kalesh that seem to be genetically distinct from everybody else. We know that your genes and languages seem to track together very closely, but this recent study seems to infer that there's something beyond genetics that determines human language. And what the authors here did, a couple of things they point out in terms of the variation of language across the globe in genetics. Number one, whereas I mentioned earlier, your genetic variation seems to diminish as you go further away from Africa. That's not the case with language. The variation in language is constant across the globe. And secondly, often you find in geographic isolates, what you see is that genetic variation tends to diminish. And you don't see that so much in languages where you have geographic isolates. The variation in language sometimes can actually get greater. So the point of these authors was making that there's something beyond our innate genetics driving human language. There's some other cultural aspects beyond our DNA driving this. Another paper that I came across that I thought was very appropriate for this conference is authors that looked at DNA from a burial pit in Lebanon from Crusaders from 1,000 years ago. And the interesting finding of these authors is that when they genotyped the DNA, they found 2 distinct ancestral groups. They found the Near Eastern group, the European group, so these were the European Crusaders that came on the Crusades. And obviously they were working with people from the Near Eastern group. But then they found a 3rd group, an admixed group, which is basically interbreeding between these 2 groups. And the interesting finding of this paper is this admixed group, although it was present for the bones of in this grave, is no longer seems to be present in modern-day Near Easterners. So these individuals that were a product of this interbreeding of these two groups, it seems their DNA has not been passed through the generations. And switching gears now and talking a little bit about the research I do, which is really focused on using ancestry mapping to understand disease and looking for disease genes. And the way it basically works is that we look for an admixed population, a population that has distinctly two different parental populations, and then we genotype that population using genetic markers that are genetically distinct between the two parental populations. And then, as I showed you in that printout from 23andMe, what we basically look for is the patchwork of DNA from these two parental populations. And sampling individuals with and without disease, we look for higher levels of ancestry in those individuals that have a certain condition. So to illustrate this, I have a little diagram here. So if you see two different populations here, one with the blue DNA and one with the red DNA, and let's say the ones with the red DNA have some certain disease locus that we're trying to map. So the first generation of interbreeding with these two, you're going to have just one separate chromosome from each parental population. But in subsequent generations, what you'll find is that those parental segments from the two distinct populations will start to break down into smaller and smaller segments. And we can actually infer the length or time from admixture based on the length of these segments. And if that disease gene continued to migrate on that red background, now when we sample individuals, but the black dots are symbolizing people with the condition we're interested in, they're typically going to have more often that ancestral background from their parents with the red DNA. And so I couldn't give a talk in a group that I know is populated with a lot of physicists without showing at least one formula. So here it is. Enjoy. This is a formula geneticists use basically to compute the diversity, allelic diversity in a certain locus between two parental populations. And it's key in doing ancestry mapping because it's not only any genetic locus that we want to use. We want to use genes or a variable polymorphic loci that have great diversity between the two parental populations, and the more diversity, the better. So the research I do, it focuses on African American populations, and most of you probably know our African Americans are descendants of people that were brought here on the slave trade from Africa 300 to 400 years ago, and came through mainly the southern United States. So about 100 years ago, most African American individuals were in the southeast United States, but over time, they're dispersed across the United States. And so the modern day African American individual has roughly about 20% European DNA, and this varies widely across the United States. There's some areas in the southeast part of the United States where you have individuals with over 90% African ancestry, but then there's other areas where it's almost 50-50. And that's actually the ideal proportions to do ancestry mapping, having that even distribution of your two parental populations. Here's some example of some work that published looking at prostate cancer, which is increased prevalence in African American populations. And so on the right here is a printout of the proportion of African ancestry that's either over or underrepresented in men with prostate cancer. And so what we're looking for here is actually peaks where there's a higher than expected or lower than expected ancestry as possible areas that harbor disease genes. And so the example I give here, this 5Q region has overrepresentation of African ancestry. This 8Q region is likewise, and this is actually a region that's been found by other authors, so it's an example of replication. But we can also find areas of lower than expected African ancestry where there may actually be now a gene harbored on a European ancestral background. Other work I've done in a disease called sarcoidosis is also overrepresented in African Americans, as depicted on this left graph here. There are higher incidence in African Americans, particularly African American females. This is an inflammatory disorder, mostly affecting the lungs, very chronic in younger age individuals. Here's an example of the ancestry mapping we did for this across the genome. But But you'll notice in these results, I'm just showing you the 22 autosomes, not the X chromosome. But here's the results for the X chromosome. And I want to point something out that's kind of interesting that we found that we didn't expect, but a reflection of our human nature and how that can impact ancestry. So on the left, you see the 22 autosomes. And basically what I'm plotting out here is the expected versus observed ancestry distribution across those autosomes. And it really follows closely to the identity line. On the right is the X chromosome, and that does not follow the identity line. And we were scratching our heads a little bit about why that's the case, but then we thought about the assumptions we made in our model. And one of the assumptions was that there was an equal contribution from both female and male admixture into this population. But if you know the history of admixture in the African American population, and particularly the early history, what you know is that most of the European admixture that came into the African American population was from white slaveholders mating with their black female slaves. And so you can actually see the evidence of that here now, hundreds of years later, a reflection of the darker nature of our human nature. Switching gears a bit, staying with the theme of ancestry, here's some work we did on a marker, a DNA marker that increases the risk for cancer, and we looked at the contribution, the influence of that actually with red wine consumption. And it may seem a little strange to you, but this red wine contains resveratrol, and the thought was that this may have an effect on this reaction. But we wanted to see the effect that ancestry might also have. And here's data that we did not publish, but we found very interesting because when we were looking at red wine consumption, we also looked at consumption of other alcoholic beverages. And so on the y-axis here, we have a proportion of African ancestry, which ranged from 100% to 0%. And as you look across this graph, what you'll notice is that whether or not you consume any of these alcoholic beverages tends to be related to the proportion of European genes you have. Those people with more European ancestry were more likely to consume alcoholic beverages. So an example here of how ancestry might actually affect your predisposition to consume alcohol. A more interesting finding, and this is work that I didn't do but actually got to review and then write a commentary on, is DNA methylation. And so quickly, DNA methylation is basically a methyl group attached to your DNA molecule, and they basically form the switches to turn on and off genes so that they're transcribed or not. And critical in development in terms of turning on and off genes early in life, but then later they can be turned on and off by mistake and cause cancer. And so researchers are very interested in what genes are methylated or not, and there's also genetic variation that affects ancestry. So the Native American population, as you might know, has been long exposed to tobacco, long before we have. And actually, they introduced tobacco to us, and it's been part of their religious practice, actually. And what these researchers showed was that certain genetic variation that affected methylation in lung cells that increased or decreased risk for lung cancer was different in Native American populations. Basically, the Native Americans had the genetic variation that predisposed less to methylation of these genes, turning them on to be carcinogenic. And so here's an example where a religious practice might have actually influenced the genetic variation here in lowering the risk for lung cancer. So I want to close, and I'm not going to belabor this because you heard from the last speaker, but just the idea of the Neanderthal genome and how it actually might be intermixed with the human genome, or is. I mean, we've seen, there's evidence of that, and these authors have used fossils to genotype some Neanderthals and show the closeness of it to humans. But the point I want to make here is now this data or information has now been brought back into the modern ancestry companies, and so they will tell you about your Neanderthal ancestry. And so here's an example. This is not my DNA, just so I want my family to know I was not in first place, but somebody has to be in first place, right? And so they will tell you the proportion of your Neanderthal genome and how that relates to both your friends and family as well as all the people that do their genotyping. So very interesting. So I want to close on a more serious note, and I want to recognize another prominent Catholic geneticist, a contemporary of ours, and his idea of what it meant to be human. And so Jerome Lejeune, who is now considered the servant of God, he was discoverer of trisomy 21, which we know causes Down syndrome. And Dr. Lejeune really loved his patients and wanted to do something for them, and it was driven by this discovery to do something well. And it struck him terribly to find that they were using his information in the way of actually destroying human life. He was recognized by the American Society of Human Genetics in 1969 with his highest award, the William Allen Award, and he gave an address at that meeting which basically changed his scientific career forever. During that address, Dr. Lejeune called out those individuals who were using his discoveries to snuff out human life and the tragedy of that. And that resulted in him being ostracized from the scientific community the rest of his life. He was persona non grata. But he had that podium, that stage, and he really wanted to make known the importance of this in furthering human life, not snuffing it out. And so I want to close with a quote from Dr. Lejeune's address, which I think is really appropriate for this conference today. And so he was talking about when does life begin, but he switched the tables a little bit. And so, and quote, "For the sake of simplification, we have devoted ourselves to the particular query, when does man begin? It appeared to be quite a clear-cut question, but should we not have been better inspired to start with the necessary question, what is a man? No dictionary and even no legislation or any country or civilization has ever fully answered this question. Depending upon what different specialists look for in a man, his quality, his individuality, his molecules, or his internal or external life, the technician, the personalist, the information specialist, and the pragmatist have given their own answers, not to the question we ask them, when does man begin, but to the unspelled challenge, what is a man? Certainly, this is the crux of the matter, but such a crux is possibly too heavy to be carried by a human mind. And so with that, I close and thank you for your attention, and just ponder Dr. Lejeune's words as we travel through this conference. Thank you, everybody.